Good morning, my dear students. Today we're discussing about Human Genome Project. So, what is the Human Genome Project, and what is the importance? What is the goals? What is the advantages? And then, what is the importance of the Human Genome Project? Now we discuss. So, simply, Human Genome Project is a organism complete study about the simply study about the DNA. Study about the DNA is known as the Human Genome Project. Human Genome Project is a complete study about the DNA only. Complete study about the DNA is known as the Human Genome Project. So it including what is the DNA? DNA containing the genetic material. DNA containing the genetic material and nitrogen base pairs, adenine, guanine, thymine, cytosine, uracil. These type of nitrogen base pairs are present in the human body and as well as the other living organisms also. So here it is including all of its genes, and each genome containing the all the information need build up the maintain the organism. Each and every gene it containing the some amount of information. That information it is a build the one organism. So one type of, for example, particular A gene, for example, 1, 2, 3, 4, 5 several genes are present in the human body. So one for number one gene is related to the heart, second number two gene related to the brain, and etc etc. Each and every organism it contains the particular types of genes. That particular type of genes it containing the information about the a particular type of organism. So here a copy of entire genome, more than how many genes? More than 3 billion genes, more than 3 billion billion DNA base pairs containing the contained in cell have the nucleus. And Human Genome Project was the international research effort that determine the sequence of the human genome and identify the gene it contains. And in here total how many DNAs? Total 3 billion DNAs. In each and every DNA it contains the lot of genes. So what is the sequence? So number 1, 2, 3, 4, 5 sequence, or A, B, C, D alphabetical sequence. So one sequence of genes are present in the one organism. That sequence is also identified by the Human Genome Project. And the next one here, Human Genome Project is coordinated by the International Institute of Health and U.S. Department of Energy. These two departments coordinate the Human Genome Project. So HGP. So here additional contribution include the universities across the United States, international partners. So international partners like United Kingdom, France, Germany, Japan, China. These countries are involved in the Human Genome Project. So Human Genome Project is starts in the 1990. It starts in the 1990. Was completed in the 2003. Total 13 years are occurred in the determination of the gene sequence. So the 2 years ahead the original schedule, and then the work of Human Genome Project allowed to the research to be to understand the blueprint of the building of a person and learn more about the functions of the gene and proteins and different types of fields like medicine, biotechnology, and life sciences, and etc fields. So in here 30 years field is a determination of the gene sequence, and this gene sequences occur in the different types of subjects like biotechnology and etc. So and identify the human sequences also, and due to the widespread of the international cooperation advised to field the genomic. And genomic mean especially sequence analysis. Sequence analysis means determination. Sequence analysis means one type of gene it containing the how many nucleotides and what is the functions, and second type and third type, fourth type, fifth type. The sequence is determined. And the next one and sequence analysis, as well as the major advance in the computing technology, and rough draft genome was is finished in 2000. So rough draft is finished in 2000. It is published in the 2003. And the next one here, the first available rough draft assembly genome completed by the Genome Bioinformatics Group of University California. So the California, first of all, the rough draft completed by the California University and Santa Cruz, and these different types of different types of universities are involved in there. Ongoing sequence led to the announcement of the essential complete of the genome in April 2003. but in 2000 the rough draft it completed, and 2003 it is the officially announced the total human genome project in 2003. And 2 years earlier than the planet, in May 2006, another milestone was passed on way, completion of the project, when sequence of the last chromosomes published in the journal Nature. Last chromosome published in the 2006. So 2003 it is the first one published, and second, third, fourth, fifth number of publications are occurred about the human genome project, and the last publication of chromosomes about in the particular information in the year of the 2006. And the next one, and the major goals. What is the goals of the human genome project? Human genome project is the goals is the first of all, what is the goal of the human genome project? So human genome project is a different types of goals are occurred. So what are there? Like the project where provide the complete and accurate sequence of the 3 billion DNAs. In 3 billion DNAs, which one is the first one, which one is the last one, which one is the middle one? Is the total sequence is determined by the human genome project. The accurate values are determined with the human genome project of the 3 billion DNA base pairs is the make up the human genome project. And to find all the estimation of the about the human body, also 20,000 to 25,000 human genes also estimated. And the next one here, 20 to 25,000 human genomes and 3 billion DNA base pair sequences. And the project also aimed the sequence of the genome of several other organisms that are important to medical research, and such importance the medical sequences involved in the mouse, and the next one rabbits, rats, fruit fly, and etc. Live organisms also involved in here. And in addition to sequence of DNA, human genome project is a sort of to develop the new tools, obtain the analyze the data, and to make its information widely available. So the next one, the human genome project is declared and completed in the 2003, and initially rough draft was human genome available in the 2000 and February 2001. And the working draft has been completed in the published, followed by the sequence of the mapping human genome on the complete information about the human genome project available at the April 14th 2003 onwards. We are study about the each and everything of the human genome project. Officially, unofficially 2000 completed, but officially in 2003 April 14th is completed the human genome project about the including human body, in not only living organisms, including the human body. Identify the 22 to 25,000 genes are officially announced by the human genome project. And the next one, although the spread the 99% of the human genes, and with the 99.99 percentage accuracy, major quality assessments of the human genome sequence was published in the May 27 2004. So especially human genes 99.99 percentage accurate gene sequence is published in by 2004 April 14th. And the next one, indicating over 92% sampling executed in the 99.9% accuracy is within the intended goal. And the addition of the human genome project is sequenced the genome several other organisms, including beaver's yeast and roundworm and fruit fly. Means not only human body, these type of genes are present in the which type of live living organisms. That one is also studied. And then in 2002, researchers announced that they had also completed the working draft of the mouse genome also in 2002. By studying of the similarities and differences between the human genes, those organisms, and research can discover the functions of the particular gene, identify the which genes are critical for life. So not only study about the genes and compare with the human body, mouse genes compared with the human body, and different types of living organisms compared with the human body, which one is related, which one is very critical for life surviving purpose, that one is also studied in the 2002. And the next one, what genome donors. So next we will discuss about the human genome project, study about the human donors also. What is the human donors, and which one, what is the importance of the human donors, now we discuss. So in international human genome sequence consortium, means IHGSC. So international public sector human genome project researchers collected the blood sample from the female and sperm from the male, some from the male from a large of the donors. Large of the donors means number of members are involved in the donors, and collected the blood sample from the collect the blood sample from the female and from sperm from the male. These two samples are collected from large group of members, and only few many collected samples are processed to DNA resources. The donors identified were protected so their donors or not scientists could know DNA sequence. And DNA clones for many different libraries used the overall project. Different types of libraries are laboratories libraries are determination of the DNA clones, and with the most of those libraries being created by the Dr. Peter and DJ Jong, much of the sequence 70 percentage, greater than 70 percentage reference genome produced by the public HGP means Human Genome Project came from the single anonymous male donor from Buffalo, from the Buffalo only. And the next one, human genome scientists are human genome project scientists were blood cells from the blood of two male and two female donors. So collect the blood sample from the two female and two male donors, randomly selected the 20 of each. So in 20 members, randomly collect the two from female, two from male, and collect the blood sample. So here the donor yielding the separate of the DNA library, and most of these libraries means RP11 was used considered by the more others due to the quality consideration. One minor technical issue is the male sample contain just of DNA. Sex chromosomes means X and Y chromosomes is presented in the male candidate, and X chromosomes only presented in the female candidate, and other 22 pairs of chromosomes is autosomes. So autosomes and body related chromosomes. So body related chromosomes, how many is present in the human body means 22 pairs of chromosomes, and two chromosomes are sexual related chromosomes, that is the XY chromosomes. So the XY chromosomes are present in the male, and XX chromosomes is present in the female. This one is determined by the with the using of the human genome project. And major findings of the human genome project, what is the major findings? So human genomes is consisting the above 300 crores nucleotide bases are determined, and a large gene is contains 2.4 million nucleotide bases. So in worldwide the large gene containing the how many nucleotide bases means 2.4 million nucleotide bases are presented in the large gene. So total how many nucleotide bases are identified? Above 300 crores nucleotide bases are identified. In here 2.4 million nucleotide bases are presented in the large gene, that is the dystrophin gene. The dystrophin gene is containing the how many nucleotides? Two nucleotide bases mean 2.4 million bases. And 99% of the nucleotide base are the identical all people. 99% is accuracy values are identified. The human genome containing about the 300 genes. So human body it contains the how many genes? 30,000 genes are presented. So applications. So what is applications? So applications is a potential benefit sequence to the human genome expand to the many fields from the molecular medicine, better understanding the human evolution, purpose. So these are identified. These are the applications. So human genome project is a sequence that DNA can help the understanding the disease including gene type, and the next one treatment, which type of drugs are involved in here, which type of diseases occurs in the body, how to form the disease. So this disease came from which type of animal. So these are all are studied with the using of the human genome project. And the next one is another proposed benefits to the commercial development of the genome researchers related to the DNA based products and multi-billion dollar industries. So here development of the gene related products and sequence of the DNA stored in the database available anyone on the internet. The US National Center of the Biotechnology Information and House of the Gene Sequences database database means gene bank. Sequence is known the hypothetical genes and proteins and etc etc. And study about the genes, total information available in the total available in the USA National Center of Biotechnology Information. And other organisms are genome bioinformatics groups, universities, California, Santa Cruz, and etc etc. Universities also involved in here, searching it for the especially gene information. And computer programs has been developed, it depends upon the gene, and processing identify the boundaries between the gene and other features, and raw DNA sequence genomes and others, and identify them gene sequences, advantages, disadvantages, how to form the disease, which type of disease, and what is the treatment. These are all are studied with the using of the human genome project. And advantages, finally advantages. So advantages of the human genome project is especially knowledge of effects variation of the DNA among individuals, and revolution of the diagnosis and treatment, prevent the number of diseases in the human body. That is the main advantage, and provide the clues of the understanding the human biology. And human biology is understanding purpose also study about them. These type of projects means human genome project. So this is about our topic. If you have any doubts from this topic, tomorrow we will discuss. Thank you.